Okay.
So we'll get going. So welcome everyone. This is our monthly AKCI lecture series, and also we're inviting the KCA mentor program sort of into this. I think we'll have some more people join in the next few minutes, and we're excited to sort of kick off 2025 with Dr. Joseph Bonventre. You see his titles there: Chief of the Renal Division and Engineering and Medicine Division at Brigham and Women's and Harvard Medical School. And this was Vincent's suggestion. We asked him to give this talk on KIM-1, obviously a lot of emerging data that Vincent and others have been involved in around kidney cancer. We think very exciting and a potential biomarker. So we'd all love to get smarter about this. So, Dr. Bonaventure, we really appreciate your time and expertise, and the floor is yours.
Okay. Well, it's a pleasure to be here. I hope I'm visible and you can.
Yep, all set.
My headshot is okay. I can't actually see it here, but
yeah, it's good. It's good.
Okay. Sometimes I see these talks with person's head halfway seen it. Okay, so I'm going to talk about really my favorite molecule, kidney injury molecule 1, and I'm going to focus on a number of components, but primarily provide data on its role as a biomarker of kidney injury and renal cell cancer. These are some disclosures which primarily relate to actually kidney safety. Many of the advice opportunities relate to dealing with safety, but also with regard to developing new therapeutics. Okay, let's see. Yeah, so here's my outline, basically. So I'll talk about KIM-1 as a biomarker of kidney injury and a prognostic biomarker for chronic kidney disease progression. And then I'll talk about it as a prognostic biomarker, a little bit of diagnostic. I won't touch on really I'll talk about it as a predictive marker, but really focusing on prognostic and to some extent diagnostic in the context of renal cancer. And then I'll talk a little bit about the functional properties of KIM-1 relevant to injury and cancer. And this is an area that we've been over the years involved with to try to look at mechanism and try to relate it to some of the clinical observations. So in the beginning, this is now, mid-1990s, we were interested in looking for molecules that would represent new targets for, in particular, acute kidney injury. And at the time, it was just at the time of mRNA chips, and we wanted to do a differential screen. Fortunately, at the time, we didn't use that current technology, but we used another technology, which was a representational difference analysis technology, which is an iterative procedure whereby you isolate messenger RNA from two samples, one normal, one post-ischemic. You make cDNA, and then you essentially put two different types of linkers on the two different types of sources of mRNA coming from two different sources, or cDNA, and then you put it at a 10-to-1 ratio. So you put, for example, 10 times the cDNA of the normal kidney versus 1 of the cDNA of the post-ischemic kidney. And then you try to use primers to amplify, and you'll only amplify the post-ischemic cDNA if it's present in, let's say, more than 10-fold of the control. And then what you do is you do that like 3 times, go through 3 rounds, and you pick up We did here, as you can, you can see on the, let's see down here, you can see KIM-1 and these bands of KIM-1, and another one was NMB, which is an interesting molecule, but it hasn't sort of seen its day in the sun yet, but it's becoming even more interesting lately. And so then, sorry, I'm just going back and forth here with these. So then what we did was we looked at KIM-1, and to make sort of a long story short, we got particularly interested in it because it was expressed in the epithelium. At the time, we also identified NGAL, and we decided not to pursue NGAL. We decided to pursue KIM-1. NGAL was more expressed in the interstitium, and we made the conclusion that it was a sign of more inflammatory reaction in the kidney. That may or may not have been a right of decision at the time, but we focused because we were interested in the proximal tubule in particular, and KIM-1 was specific to the proximal tubule. And on the right, in the human atlas, you can sort of see, this is now more recent data, not ours, but where KIM-1 is really markedly expressed in the kidney. There's a little bit of expression. We knew right from the beginning that there was some expression in the colon. There's testis expresses it, but it's really primarily focused in expression in the kidney. And over the years, it's been confirmed by many groups that it's the molecule that's expressed more with injury in the kidney than any other molecule in the database. And you might expect that. We were looking for low-hanging fruit, molecules that were markedly upregulated, but it turned out that the one that we found, which we named then kidney injury molecule 1, has consistently been confirmed to be the molecule that's most upregulated. And so on the left-hand side here, you can see some of the cartoons where the molecule is one transmembrane molecule, and it's heavily glycosylated, and it has a Ig-like domain in the extracellular region. Early on, as another reason for studying it, we looked in the urine, and we found it was present in the urine. And we did some biology with Veronique Bailey and Michelle Sanicola, who were at Biogen at the time. And we found that it was cleaved by a metalloprotease. The specific one we didn't identify then, but we had inhibitors, and so it's cleaved and appears in the urine. It's spontaneously cleaved, at least in most cell types. We discovered some antibodies that could prevent the cleavage, which we're currently still using. And we also established some antibodies that would pick up the ectodomain and also the intracellular domain. And then we did studies to characterize the localization. This is a human sample with acute tubular injury, and you can see the staining on the apical aspects of the proximal tubule, and no staining in the glomerulus. And here, fast forward now 25 years later, we've convinced Sandro Santagata, who's actually a neuropathologist, but he's gotten more and more interested in the kidney. And what he's been doing is setting up a system here for looking at 10 analytes at the same time on a clinical sample. And so we asked him, well, he came to us and we talked about KIM-1, and I said I thought it would be a good thing to look at. since it's very specific, and you can see the samples here, and this is from a pathology report of minimal change disease, actually, which is characteristically difficult to diagnose on the light microscopy because there's minimal changes. It's an electron microscopy diagnosis. But here, so the histopathology report was basically, in the proximal tubules was mild tubular dilatation, focal brush border loss, and isolated mitotic cells. So there was some evidence that there was some activity, but it was not as clear as you see with KIM-1 staining quite dramatically across really most of the proximal tubules in the sample. And here's this is also an animal study. This is a rat, actually, and you can see the very nice apical expression. And this relates to, what we're going to talk about later is plasma levels of KIM-1. I've told you it appeared in the urine. We focused for many years on the urine, but it's clear that it appears in the blood, as we'll talk about. And part of the reason, which I don't show you here as clearly, but on other slides, you can see that this apical pattern becomes more diffuse when there's injury and loss of polarity of the proximal tubule. So the proximal tubule is very, very polar, has tight junctions, and separates the apical and basolateral compartments very nicely under normal circumstances. But that separation is lost with injury. The more severe injury, the more it's lost. And here you can see why we got excited about this. This was now quite a while ago. These were 1996. And you can see the staining, both RNA in situ and immunohistochemistry. This is the, basically it's primarily cortical and actually in most cases, more in animal models, more in the outer medulla, because that's where many of the animal models have most of their injury. And here's a human case, which shows the association of apical staining in green here with fibrosis staining. Actually, in this case, it's smooth muscle actin staining in the region right around these damaged tubuli. Okay, and then we showed it's upregulated very quickly. As I mentioned to Brian before, I know we want to talk a lot about renal cell cancer, but I think a lot of these observations are relevant. I mean, and I think they overlap in terms of mechanism. So it's useful to sort of think about it, I think, in the context of injury and what does injury have to do with renal cell cancer. We knew early on, actually, I would say, in the papers that we published in 95, 96, that 769P cells had really very, very high levels of endogenous KIM-1 expression. Those are renal cell cancer cell lines. And so I'll get into the cancer piece later, but you can see that even with 10 minutes of ischemia and reperfusion on the left-hand side here in an animal model, this is the mouse now, 10 minutes of ischemia, looking at 24 hours later, you can see the marked upregulation of this molecule. And then obviously, if you go 20 minutes for ischemia or 30 minutes, there's a dose response. And urinary obstruction does it, aristolochic acid does it. Essentially, this molecule is a sentinel molecule for injury in the kidney, no matter, but it doesn't distinguish one type of injury from another, except it does tell you that it's proximal tubule injury. So there are a few toxicants that, in fact, I'll mention it here. So then what we did was we were called at one point. There was a group that was putting together a consortium, or had a consortium put together for nephrotoxicity, and and this was primarily a pharmaceutical consortium. They tried about 10 or 15 biomarkers at the time, and this is a composite of studies that were done primarily at Merck and Novartis, but also a number of other companies, with dose responses and time related to with 10 different nephrotoxicants, and and so it's a little bit hard to read, but the colors relate to the grade of damage, looking at it from the perspective of the nephropathologist. And you can see with increasing time and dose, here in the context, this is really dosing here. You can see that to the right here, you can see these marked increases with almost every nephrotoxicant, except, let's say, if you look at lithium, there's no change. Well, lithium's a distal nephrotoxicant. If you look at furosemide here, which is also, oh, sorry, apologize for this, which is also a distal nephrotoxicant, you don't see any increase. Okay, so there was a lot of studies that were done, and then ultimately, describes the results that we got. And again, all the statistics was done at Merck. I basically went along for the ride to sort of explain what the properties of the molecule are. But we published this this paper which summarized all the data, and essentially this is the area underneath the curve. You're all familiar with biomarker curves, receiver operator curves, and and here's the pathology, the histology score. So this is on a scale of 0 to 3 histology. So here's all of the data where there's any histology, as histological differences relating to injury. And you can see that area underneath the curve is between 0.85 and 9 for BUN, NAG, N-acetylglucosaminidase, and serum creatinine. And but if you go to the very mild injury, 0 to 1, KIM-1 stays extremely high. It actually is about 0.95, whereas serum creatinine becomes pretty much useless. As you know, about 0.5 would be completely chance, and NAG and BUN also become really much less effective. Okay, at the same time, there was a consortium in Europe that published the same year and found that KIM-1 had an AUC of 0.99, and the other ones were clusterin and lipocalin is NGAL. And you can see what we weren't involved at all in this study. Okay, and then the FDA subsequently in 2008, actually for the first time that they came together with the EMEA, the European organization, they qualified 7 key biomarkers for nephrotoxicity in animal studies. And they also commented that they're likely to be useful, but they didn't officially qualify them for human studies. And KIM-1 was there. These are the tubular markers. Albumin, it's important to recognize, particularly those of you who are clinicians, that albuminuria is a very, very sensitive marker of nephrotoxicity or toxicity. And I mean, you're oncologists too. I think this is something that people don't appreciate. And clusterin and trephine factor 3 is not a particularly useful marker. Okay, and then since that time, or that was in 2008, the FDA published this. This is coming out of the FDA, in CEDAR, where they published a number of applications for drug development that came in using KIM-1 after they qualified the molecule. And then in 2018, it was qualified for phase 1 clinical studies to evaluate nephrotoxicity. Okay, I won't go into that. And then now going to the plasma level. So in 2014, we published the first paper on plasma KIM-1. And what we showed was that injury, again, here's the urinary KIM-1. So I showed you before the 10 minutes of injury looking at 24 hours later actually showed a good amount of histological staining, but it also shows in the urine 24 hours later, you can see it just with 10 minutes of injury. This is normalized to urinary creatinine here. You can see here that even with 10 minutes of injury, you could pick it up in the plasma. And 20 minutes of injury, you see it more. 30 minutes, you see more. Creatinine doesn't really change, but it doesn't become statistically different unless you do 30 minutes of ischemia. So we thought this was pretty interesting. And then we looked at cardiac surgery patients, ICU, the plasma levels, and showed that there was a significant increase. If you compared with controls, there was much less in controls. KIM-1 showed not unexpectedly in the urine, and the urine KIM-1 showed a correlation with plasma KIM-1. Here's CKD, and the higher the levels of KIM-1 in patients with CKD, the lower levels of estimated GFR and CKD stage. So putting it similar way, so as the CKD stage goes up, the plasma levels go up. And then we also included in this paper type 1 diabetics, and showed that the higher the, so here's the prognostic biomarker, the higher levels of KIM-1, the likelihood that you'd have an increased rate of estimated GFR loss. And if you segmented the levels of KIM-1, so we went back to samples that were quite old and found that if the levels of KIM-1 at day 0 in these samples were elevated, that the chance of survival without end-stage renal disease, again, these are type 1 diabetics, was much lower. So the KIM-1 was a prognostic biomarker for patients with diabetes and chronic kidney disease. Okay, and then there was a study in Europe of diabetics again, and they tested a large number of markers. In this case, actually 42 markers, and again found KIM-1 to be a leading marker for progression of diabetic nephropathy. And here was a study of 297 biomarkers, blood biomarkers. Again, we weren't involved in either of these studies. This is the study from Europe, and they found KIM-1 and CD27, which is a member of the TNF receptor family, to be the leading markers. And they said, this is a quote from the paper, we can see that almost all the gains in prediction are from just a few biomarkers here, CD27 and KIM-1. And that's a theme in terms of injury that's coming around now quite extensively, that TNF receptor and KIM-1 seem to be the best. If you had to pick two, in fact, we've done that with the CKD consortium. If you had to pick two, those are the best two. Okay. And then there was the relationship between plasma KIM-1 and increasing hazard ratio for progression. Steve Coca put together this, I think about, yeah, it was late 2018. But then I summarized this. So these are all the studies which have shown KIM-1 is a prognostic biomarker for progressive kidney injury and/or cardiovascular disease and/or mortality, which, if you think about it, why should it be a marker for cardiovascular? Maybe it's all through renal, but maybe there's something else interesting here, and the yellow ones relate to studies from the CKD consortium that we were involved with. We were the central lab for the consortium. And none of this, we weren't involved in any of the analyses, but we were involved in providing the consortium with the data. Okay. All right, so let's get to renal cell cancer. Okay. Okay, so there seems to, sorry. Okay, so right in the very beginning, after finding KIM-1, it seemed obvious to me that it would be increased in renal cell cancer, because the way I was thinking about KIM-1 at the time was that it's a marker for dedifferentiation of the epithelium, and not necessarily complete dedifferentiation. People use that term differently. To me, it's just basically reverting back. And a disappointment at the time was that it wasn't really very prominent in kidney development. So there was this bias in my mind at the time that that basically repair was recapitulating kidney development. And so then we looked for KIM-1, and it's expressed actually in day 6 of development, or it's really not a developmental protein. But it was, again, we looked at kidney cancer, I think maybe some animal models initially, and it was expressed, but it wasn't surprising to me. And so here you can see the staining. This is from Wan Han, who was actually on a K ward at the time, and we that it's tissue and urinary biomarker of renal cell cancer. It was pretty much other than ovarian clear cell, since I'm not going to present all the data that we have on this, but and much of it is not published actually. But other than ovarian clear cell, it's not a clear cell expression. We looked at a number of clear cell tumors, and it wasn't expressed in any of the other ones except for ovarian. And it wasn't expressed in most other tumors, although I think we had like 1 or 2 hepatic hepatomas at the time that out of 50 where it was expressed. And we have more recently found that it can be expressed actually in a lung tumor, or a line, and in a and actually in COVID we could get it expressed in the lung, in the human lung. And so it can, and there's a whole, so it can be expressed for reasons that were not entirely clear under unusual circumstances in other epithelial systems, but not to any extent like it is in kidney. And so again, this is really very, very early. We benefited from collaborators at the Dana-Farber at the time. And if you look on the bottom here, so we had 24 of 35 where we had positive staining. And then we had, so here was another thing we noted, that there seemed to be some staining around the tumor tissue. And so I was sort of interested in that. I still don't know whether this was that they were tumor cells or that they were just reflecting the ischemia associated within the adjacent tissue. But if you looked for positive staining of tumor cells and or adjacent tubular cells, 32 out of 35 of the clear cell and papillary cell, we had only a few at the time, but that's persisted. Chromophobe didn't stain. And then what we did was we had some patients before and after nephrectomy, and in fact, reassuring us that most of the KIM-1 in the urine was in fact coming from the tumor. At least the levels went way down, although in some cases they didn't disappear. And we can talk about that. There was an association we found with tumor size, little greater the size, the more KIM-1 in the urine now. And this was just some quantitation on the bottom of the pre and post nephrectomy. So pre-nephrectomy, we didn't have, in one of them, we didn't have anything in the urine. But in the others, if you look at the normalized to urinary creatinine, it completely disappeared in two of them, and it was quite low in a third, and then completely disappeared in a fourth. The paper, was turned down. Where was it? Yeah, it was turned down in Jason because we didn't have enough data. Kidney International took it, and they've benefited from citations ever since. And then also we published, this was a subsequent paper in collaboration with Henley from Stanford. This is but we were co-authors on this. And there does seem to be some expression in embryonal, about 50% of embryonal carcinoma and yolk sac tumors, but not in seminomas, not in seminomas or choriocarcinoma. So there is some, I don't know, I haven't sort of understood why this might be the case, although there's clearly a kidney is basically in development. If there's some problems that relate to the production of these kind of tumors, that might be explainable in that context. Okay, and I'm going to show some data that's not published, although we've tried for 10 years, but just for historical purposes. So we had a cohort from the Dana-Farber and a Würzburg cohort, and looked at the levels of plasma KIM-1 and related it to tissue expression and tissue mRNA levels. And you can see on the top left, the plasma KIM-1 clear cell and papillary cell was up there. Chromophobe, these levels are relatively low, and in oncocytoma. And there was a relationship with staging, as you might expect, in terms of T and M staging. And in panel E here, the post-nephrectomy with M1, there's a drop, and M0, there's also a drop, with M0 perhaps being more dramatic. And the grade, the histological grade, also related to plasma KIM-1 levels. And in terms of overall survival, this was work in collaboration with Rupo Bhatt and Venkata Sabasetti, who was in my lab at the time. And you can see survival probability. If you looked at separated them out by quartiles. Separated them out by quartiles. That the fourth quartile had a marked reduction in survivability, and the Würzburg cohort, which we had a little bit less control over. It was given to us, brought over, but in any case, it also had the same general pattern as the and on the other extreme, the first quartile in the Harvard Dana-Farber, basically had very, very good survival. Okay. And then, so I won't, we had done some other studies there, but there seems to be a dissociation in these studies. Again, this hasn't been published, with histological staining or with mRNA. And I'm not sure what that means, but nevertheless, there wasn't a good association. This was another study that this came out of the, led by Rupo, out of the Dana-Farber and collaboration. And this showed the KIM-1 predictability, again, of survival of patients with renal cell cancer. And again, the higher the levels in the plasma, the lower the survival. And this is a slide from Vincent. Here's, and here the issue is, and we're co-authors on this, but here the notion of this paper said something about prediction. And if you look back 5 years, the patients who had higher levels had more RCC. I think this is a little tricky and dangerous, and not dangerous, but there are a lot of reasons why one might have a high plasma KIM-1 level. And in the very first paper, we actually commented that it might not be a good screening marker because there's so many people out there with chronic kidney disease. Unless you really screened for those who didn't have any kidney disease and you knew that. But on the other hand, if someone has a high plasma level of KIM-1 and they don't have a good reason for it, then one could argue that it's a good reason to screen with at least an ultrasound. That's an issue which I brought up a couple of times, and that has ethical implications and stuff, and we can talk about that if you want. So here is Vincent's data, again showing that the higher the level of plasma KIM-1, the worse outcome, overall survival. This was also reflected by progression and metastases. So that, again, this general theme, which seems to be recurrent now, sort of reproducing what we have with chronic kidney disease, that when people look, it seems to be a marker for badness. And so, here I just showed the comment that from the internet, non-invasive biomarker KIM-1 is front and center in renal cell carcinoma investigators. And when Vincent was giving a talk to the local group here, and Tony was there, Kaluri, and he said, KIM-1 has become important because you can tell because so many people are trying to take credit for it, which is great, but he also, so he sent out this X poll to his people who are his followers, and of these biomarkers, he got 200 responses in an hour, I think, and so maybe some of you responded, but KIM-1 was chosen above of the four. Okay, so let me talk a little bit now of the functional properties. So, and here we could go very long, but I'm not going to, because I was asked to sort of stop in 5 minutes so we can have time for questions. But KIM-1 is a phosphatidylserine receptor. It was the first one that was, as far as we know, that was published that's not on some kind of professional phagocyte. And so it's a phosphatidylserine receptor. It's and chronic KIM-1 expression is bad. Chronic KIM-1 expression is bad, and I'll show you some data reflecting that. Acute KIM-1 expression, from my perspective, cannot be bad. It has to be good for you. If you're a kidney epithelial cell, if you're a proximal tubule cell, evolution is not going to give us a response as dramatic and make this molecule the molecule that's expressed more than any other protein in the database, if it's disadvantageous, if it's going to harm you. And a driving force in evolution is blood loss. And so it makes sense. And we have data. We have here on the bottom right here, KIM-1 mediated phagocytosis reduces acute injury to the kidney. Relevant, I think, because of what I'm going to say, relevant to thinking about it in cancer also. Here's a picture of the interaction of apoptotic cells, which obviously express phosphatidylserine. Renal cell cancer also expresses phosphatidylserine. And here you can see the red in the bottom left-hand part here, the red KIM-1 binding to here, some of the binding, but you see the apoptotic cells that are in green. Here's the sort of intercortical outer medulla staining of KIM-1. And here is another property. We have this paper in EMBO Journal, Craig Brooks was the first author on, and it shows basically a phagocytosis that we think is also, as we develop this, related to an immune response. So first, here I'll just show that in the context of acute injury to the kidney, and kidney epithelial cell being the major effector of the injury response, we generated with Vijay Kutru a mutant mouse that lacks the mucin domain of KIM-1. And in fact, if looking at it in serum creatinine, after ischemia reperfusion, there's a more of an elevation in serum creatinine in the delta mucin mouse, the mutant mouse. And the tubular necrosis score correlates with that. And this was a whole paper. So I won't go into the details. The other piece, when we overexpress KIM-1 in transgenic, and this was Ben Humphreys when he was here did this, we actually got chronic kidney disease, and the animals died relatively rapidly, and we got a nice model for chronic kidney disease, and including cardiac hypertrophy and anemia. So we think that KIM-1 is good for you acutely. and but chronically, it's bad for you. Here's the size of the kidney. Here's what it looks like. Here's the histology. And another thing, we recently published a paper in Cell Metabolism that I'll just show this slide from. And we have the notion that, in fact, there's a KIM-1 sitting on the surface of cells, proximal tubule cells again, can take up albumin if it's linked to palmitic acid, so long-chain fatty acid. Doesn't seem to do it if it's oleic acid. And this relates, may relate to the yin-yang of diabetes and diabetic, where oleic acid is good for you and palmitic acid is bad for you. But in any case, we believe that KIM-1, relating to this chronic injury, takes up albumin, fatty acids, overloads the mitochondria, relates to an increase in reactive oxygen species, DNA damage, cell cycle arrest, senescence, and fibrosis. Also activates the inflammasomes. Okay, and it's expressed in, this is redundant now, just it's clearly expressed in diabetic patients. And this is a slide this is a tongue-in-cheek slide. Let me just, okay. So this is a paper by a very, very good group, one of the leading groups in the country right now, and led by Catalin Sustek at Penn, good friend, who published this paper in Nature Genetics with really one of the enormous amount of work with multi-omic analysis and spatial profiling. But it's interesting, one of the main conclusions upon careful, top right, subclustering of our COSMICS data, the spatial transcriptome, and we noted VCAM expression in both PEC's parietal epithelial cells and injured proximal tubule cells, while HAVCR1, which is another name for KIM-1, hepatitis A viral cellular receptor, was specific for injured proximal tubule cells. So I joked with her about, and this is this slide that I've already shown you from 1996. So what goes around comes around again. So, but it's good. So it's become extremely, it's survived the movement to single cell data to be a very good marker among, again, a database analysis looked at a different way for an injured proximal tubule cell. And then this is the paper, again, that was published a number of years ago. And this, I think maybe just reading the last sentence. So in addition to participating in restoring a functional renal epithelium, KIM-1 might play an important role in the immune response. And ultimately, that's why I think that all of this is related. Here we proposed that in 2002. And in this paper that we published with Craig, this is a model. It's very naive in a certain way, but we had a good deal of data to suggest that once KIM-1 mediates the phagocytosis, it modulates inflammation and demonstrated at that time that it was important for moving phagocytosed material to MHC and MHC1 and 2, and it seemed to downregulate CD4 cells and upregulate Tregs. Again, simplistic from the point of view of a non-immunologist, but I'm learning a lot more, trying to learn a lot more immunology these days. So that may be, that, and so this is my next to last slide. So KIM-1, a diamond with many facets. All right, let me, prognostic, diagnostic, and predictive biomarker. You can imagine it might be something useful in terms of drug delivery, imaging. There are some other people who have published some papers on both of these. The consequences, it's protective for AKI, contributes to injury in CKD. There's immunomodulation and relationship to tumor genesis. It's on 5Q, and there are a number of polymorphisms which we're exploring in another context in terms of injury, which I don't have time to go into. So with that, this is a group picture of a few years back. I'll point out that certainly in the renal cell work, Wilpu and Vincent, clearly doing a lot of work with Vincent right now, and David McDermott and Tony a little in terms of co-authoring, and here other people. This is really just a small fraction of the people that have been involved over the years. So with that, thank you. Thanks very much.
Yeah, that was great. We appreciate that sort of thorough review. It gives us a nice background for KIM-1. We have a few minutes left, maybe 5 or so. I'll open it up for questions from anyone.
Yeah,
So
thanks, Joe, so much.
Okay.
Yeah, Joe, that was really amazing. I think, every time I listen, I'm kind of enthralled by how this has been like a 28+ year story
Right.
for KIM-1 starting in your lab. And many people on this call are kind of involved in this story. Brian Reaney, Dr. Reaney, led the iMotion 10 analysis in the adjuvant trial, which you cited, and he was senior author on that, led by Laurence as first author. And then actually Pratik Khanna, who's here, he was on the 2018 KIM-1 paper with Ghislaine Silo as the first author, and Rupal Bhatt and you. So lots of people have been part of this story. I think it's really fascinating, and I think there's a lot that we still need to understand about the immune role of KIM-1, as you pointed out. And a lot of this work, has been also worked on by other folks, including Dr. Freeman, who's going to give a lecture soon. But clearly KIM-1, RCC is an immune responsive disease. KIM-1 is important to RCC biology, and I think the immune roles, still have to be worked out.
Yep.
Yeah, I was gonna ask maybe a follow-on question about sort of mechanism. I think maybe third to last slide kind of, I think, was sort of hinting at maybe how KIM-1 would mechanistically be related to the immune response, but it sort of seemed opposite, right? Because you said it upregulated Tregs and downregulates activated T cells, if I got that correct.
Well,
So
yeah, but but you're assuming, I think, in your question that KIM-1 expression. So no, I don't think it's up. So if KIM-1 expression downregulates the attack response to the cell, then that would be consistent with an upregulation of Tregs and downregulation of CD4, CD8 activated. So in other words, if you think about it, again, going back to the injury piece, if it's beneficial in acute injury, our simplistic notion is that there are two ways it's beneficial. First of all, it clears out the lumen of the tubule because it binds phosphatidylserine, it binds the debris, which has a lot of exposed phosphatidylserine. But we also think, and we said in that paper, that it downregulates the inflammatory response. So if it's downregulating the inflammatory response, then it's making the tumor more tolerant, right? That means basically going to continue to grow. It won't be attacked. Again, I'm sort of on thin ice here, but isn't that right? I mean, what is that? that's the way I've sort of been thinking about it, that it downregulates the attack response from the cellular damage response.
Right. Yeah, I'm just trying to understand in light of some of the cancer data, Vincent. I don't know if you have insight here into mechanistically.
Yeah, I think, you know, the clinical data in RCC, it's always a bit confounded by the prognosis of patients, right? So Brian, as you showed, patients with high KIM-1 after nephrectomy have benefit from immunotherapy, and so clearly, you know, having high KIM-1 doesn't rule out benefit from immunotherapy in kidney cancer patients. That's certainly clear. We also know, though, that in the metastatic setting, high KIM-1 patients do worse on immunotherapy overall, but and both those may be just associated with tumor burden, right? So the KIM-1 is strongly associated with tumor burden in RCC, and high KIM-1, I think, first and foremost indicates more cancer, which explains both the adjuvant benefit and the metastatic prognostic part. But the question, which I think gets washed out a little bit in the clinical data, is what is the actual immune function of KIM-1? You know, KIM-1 is a phosphatidylserine receptor, as Dr. Bonventre discovered, and KIM-1 is necessary for facultative phagocytosis of injured renal epithelium, and that may happen in RCC. You know, I think, what's the immune significance of that with RCC antigen presentation? I'm not sure we know.
Yeah, thanks. Other quick questions before
This is
we
a
wrap
great
up?
talk.
Yeah.
Yeah, this is Ritesh. I guess one thing that's always struck me with, especially KIM-1, with a lot of the data is as you mentioned, there's so many different aspects that can sort of impact, I guess, KIM-1, right, in this space in terms of like kidney injury that's not necessarily, let's say, tumor related and things like that. Has there been a investigation, maybe Vincent, you'll comment too, but has there been like an investigation in terms of like measuring KIM-1 levels, and particularly with some of the agents like we give like TKIs, is there a sense that patients may be at risk if they have high KIM-1 in terms of proteinuria, or there like kidney damage that might actually happen naturally? And the second question I have is investigating the detection of KIM-1 in other compartments, particularly, let's say, patients with brain mets, where you have, let's say, CSF sampled, which is very, very clean, almost environment. If you detected KIM-1 expression, would you worry that there's actually like viable disease there or something like that in that context with it's such like a clean system compared to, let's say, plasma or urine?
Yeah, Ritesh, that's a great question. So there's a couple of pieces of data. So the CSF, I agree. I mean, if there's KIM-1 in the CSF, it's probably bad. It's probably coming from RCC, I think. As far as the kidney injury biomarkers, so we have an abstract at GU ASCO this year. Claire Steiner is the first author, where we adjust for like a whole bunch of kidney injury biomarkers simultaneously measured, including TNF receptor and the other ones that Dr. Bonventre mentioned. So, you know, at least in metastatic RCC, the kidney injury part is completely washed out by the sheer bulk of the tumor. And so, you know, the circulating KIM-1 in metastatic RCCs is probably over 90% coming from the tumor. Whereas in patients who don't have kidney cancer, you know, the circulating KIM-1 is coming mostly from injured kidney. So I think both are true. I think in someone who's NED after nephrectomy, if they have ischemic kidney injury, you worry about the KIM-1 not actually being a recurrence, but actually from ischemia. But in metastatic patients, I think that's really less of a concern. And in all the metastatic cohorts we've looked at, there's no correlation at all between the circulating KIM-1 and renal function or EGFR.
Oh, interesting, because I was about to say you could almost extrapolate that, you know, beyond kidney in terms of, you know, patients who are, let's say, borderline cisplatin eligible on the bladder side, where you're worried about, you know, potential toxicity to the kidney, and could you unmask maybe a more sensitive metric than, let's say, just creatinine or GFR in terms of potential nephrotoxicity to certain cancer therapies?
Yeah, I think so. So Shruti Gupta, who's in Dr. Bonventre's group in the renal division, is working on that in bladder. Cisplatin.
Got it. Okay.
Yeah, I mean, I think, well, you know, the cisplatin, I don't know if this is, so a number of years ago at the time when the FDA was interested in qualifying, and the FDA has actually been interested in KIM-1 actually, and been a little bit frustrated that it hasn't been expanded in terms of utility. But there was a study done in terms of qualifying KIM-1 for nephrotoxicity in humans, and to some extent, if you think about that, it may seem simple, but it's not if you're going to qualify, because what you're doing is they took patients who were treated with cisplatin for head and neck tumor at the Dana-Farber here, and then with gentamicin down in Texas, and they actually used not creatinine, they used exposure as a way to compare biomarkers, and it was based on that study with cisplatin that they said they'd be, so what it was that in all of the patients who developed an increase in KIM-1 in the urine, 99% of them or 98% of them had been exposed to KIM-1 to cisplatin rather than head and neck irradiation. That was the comparison. And so because you couldn't use creatinine as the marker, as the gold standard, because you were trying to replace creatinine with a better gold standard.
Gotcha. Yeah.
You see? So therefore, the study's been done. It's on the internet. I don't know if it's in the literature yet because it's taken a lot of time. I wasn't involved with it because, the FDA at the time said it's probably better for me not to be involved. So, but Sus Wykar, who was in my division, basically did the cisplatin piece, and but I just don't know that if it's gotten into press yet.
Thanks.
Very interesting.
Thanks,
Okay.
everyone.
Thanks.
Yeah, I think we're gonna wrap up just in the interest of time. But Dr.
Okay.
Bonventre, really appreciate your time and
Okay.
expertise. Very
Well,
much appreciated. Thanks,
pleasure.
everyone.
Pleasure. Thank you.
We'll see all of you real soon.
Okay. Bye-bye.
Thank you.
Bye-bye.
See you.
Bye.
Bye.
Thank you.